Friday. Hold on to your hats because now we're getting into neuronal physiology, all the details of these charged particles moving, etc. So keep track of this. This is important because, like we said, nervous system, while complex, if you get down to the basic functions of it, it's about sending signals, right? Controlling and coordinating. And so at the individual cellular level, this is the key information. Much like when we were looking at the muscular system and we were talking about how a muscle does its one job of contracting, right? The sliding filaments, etc. This is how these excitable cells are going to do their one job as well, right? So sending and receiving signals. So let's take a look. So we're going to be starting with our graded potentials. Now, we're going to spend more time on action potentials, but our graded potentials are going to be the thing that we're contrasting with. So this is still changes in membrane potential. And what we find is that graded potentials are short-lived, localized changes. And what we're going to see is that the stronger the stimulus, the stronger we go bzz, 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 bzz on the cell, the more that voltage is going to change. And the farther that current is going to flow. So we'll see, it's kind of like, and I have a picture I think coming up. You can see like that. So it's kind of like when we drop a rock into the water, right? We have the biggest effect right at the impact of the rock, and then we have this flow, outward flow and disturbance that goes from it. And as we get further from it, it gets smaller and smaller and smaller. But the bigger the rock, the more change we have right there, and the farther out it goes. Okay, so what's going to happen here is the cell is going to get triggered, right? And so what we're going to see is we're going to see how that membrane potential is going to change. So usually what's going to happen here, think of what's happening at that junction, at the synapse, right? So for instance, when we were talking about the neuromuscular junction, or even one neuron talking to another neuron, right? A presynaptic cell and a postsynaptic cell, this one before the synapse or gap, this one after the synapse or gap, right? We've got to talk across that gap, so we release neurotransmitters. And as those neurotransmitters diffuse across the gap, they're going to bind to our chemically or ligand gated channels on the postsynaptic cell. And we saw that chemically gated channels, once they bind their particular chemical, as long as it fits, those channels are going to open up. And the more chemicals we rain down on it, the more stimulating the cell, the more of those channels are going to open up, the more usually we're going to have sodium, if we're exciting the cell, sodium channels opening up, more sodium diffusing into the cell. So what we have is if this is our neuron, multipolar neuron, here's my axon, here are my dendrites, here's my soma or cell body. These are the places here where we're going to get graded potentials, right? We're going to have some cell raining down neurotransmitters on it, and they're going to receive it, and they're going to start opening up sodium channels if they're excitatory, or even potassium channels if they're inhibitory. We'll talk about that later on. This is where we're going to be hearing that signal, though. So this is where we're going to be generating those graded potentials. And the more we stimulate this cell, the more chance it's going to say, okay, I get it. I'm going to send a signal of my own. I'll relay that signal, and I'm going to send it down my own axon. Okay, so graded potentials are going to be short-lived and kind of spatially restricted. They usually occur at the dendrites and the soma or the cell body, right? This is how we're receiving the signal. And it often involves those chemically gated channels. Okay. So this shows kind of a generic stimulation, right? But at the point at which we stimulate the cell, we're going to get changes in permeability, right? Like sodium channels opening. And as we change the permeability, if we change it enough and the membrane potential changes, you might start to recruit some of those voltage gated channels as well. So that means that channels that are right next to it, that might be voltage gated channels, might start being affected. And as they're affected, they also may start allowing these different ions to move across. And that changes the membrane potential as well. Okay. So those are graded potentials. Oftentimes where we're receiving the signal, and oftentimes also due to chemically gated channels. Now we move to action potentials. So action potentials are the way that we are going to transmit or send a signal instead. Remember, it's going to start at the axon hillock, where the axon connects to the neuron, right? So that's where we're going to generate that action potential, and then it's going to quickly go down the axon itself. And these ones are basically going really fast, and they're not decaying. So rather than that ripple effect where we see it getting smaller and smaller, these ones are propagated or continuing down the axon undiminished. So we'll see that coming as well. All right, so these are the nerve impulses that our neurons are going to be sending. So it's kind of like a wave, right? So that's what we see through here, like at a stadium or whatever. We see each section influences the next section that becomes excited, and then we see changes, right? And then that continues around and around and around. So as that goes through the stadium, of course, it'll decay through time. Doesn't happen with action potentials. Okay, so remember, if we're starting at the resting state here, that's what we see down at the bottom here, number 1. The resting state is where we're on the flat, right, like that, okay? And so what we have there is we have leak channels that are open. We have sodium-potassium exchangers that are doing their work. But these other channels that are going to be involved in action potentials are just sitting there. The gated channels are just sitting there. Okay? So we can see how they have little gates. And we can also see how we have separate channels. We have sodium channels and potassium channels. Different channels for different ions, and they both have their gates closed. So we get no passage of ions through these. Leak channels, yes. Gated channels, no. All right, so that's the resting state. So like we said, all of the gated sodium and potassium channels are closed. And so what we find is that in some cases, even when we look at the sodium channel, it actually has 2 different types of gates, if you will. It's got an activation gate that we see here. It even has an inactivation gate, a second one that the potassium channel doesn't have. That'll come into play later. Okay, so step 2, depolarization. So what we're going to have here is, for instance, we're getting enough stimulation out here at the cell that is becoming more and more depolarized with its graded potentials, and that axon hillock is also becoming more and more depolarized. So what's going to happen here is that we're going to have these voltage gated channels open up. So a depolarization, the sodium channels are going to start to open up. So there's a little lead-in right here that's not really reflected on the graph. As it's becoming more depolarized like we see there, that's due to our graded potentials, right? Stimulating the cell, more and more of these chemically gated channels are opening, more of the sodium is piling into the cell, it's making it less and less negative. At some point, it's going to reach what's called threshold. So at threshold, kind of like if your little brother's bugging you, what are you doing? 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 Each little poke builds and builds and builds, and at some time you lose your cool and there's this big explosion, right? That's what happens with the action potential. So with an action potential at a certain voltage, which is called threshold, which is about at negative 55 millivolts, then these voltage gated sodium channels are going to open up. So the gates are going to open and we're going to have this flood of sodium going into the cell. And that's what we see here. With all those positively charged sodium ions going into the cell, we see a very quick depolarization. So like we said, those voltage gated channels are going to open up at what's called threshold, which is generally about negative 55 millivolts. Okay? So that's going to be important, right? And this is actually positive feedback, because what happens here is as that becomes more less negative on the inside of the cell, then more voltage gated channels are going to open up, which makes more sodium flush into the cell, which makes more of these channels open up. So that's why it's really, really quick. Okay. Second thing, we've done this big depolarization here, right? We started at about negative 55 millivolts, which is where these voltage gated sodium channels opened. Now here we see a turnaround point. At that point right there, what's going to happen is our voltage gated sodium channels are going to get the close signal. So this is about plus 30 millivolts. At about plus 30 millivolts, our sodium channels, which were open, get the close signal, and now our potassium channels open. So at plus 30 millivolts, 2 things are happening. The sodium channels are closing, and now the potassium channels are opening. Now remember, potassium we have a lot of potassium inside of the cell, positively charged potassium inside the cell. So if we open up the gates, that positively charged potassium is going to start being lost from the cell. It's going to be the inside of the cell more and more and more negative. It's going to repolarize the cell, right? Going to make a difference across that membrane potential again. So sodium channels close, potassium channels open. These are voltage gated. Like we said, this is about plus 30 millivolts. Ooh. Excuse me. Sorry. Okay. I know you said bless me out there. I felt it. Okay. So once we get down towards resting membrane potential, what's going to happen is our potassium channels are going to close, and the inactivation gates that we see here on our sodium channels are going to reset as well. Now it takes a little bit of time for all these different gates to close for the potassium channels, so we get this overshoot that's right there, which we said is hyperpolarization. So it takes a while. I mean, like if you were running down a hallway and you had to close all these open doors, it would take you a while to go bam, 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 right? Same thing with these potassium channels. Voltage gated potassium channels, as we reach resting membrane potential, they're going to close, but it takes a while for that to happen. So we get that little bump that we call hyperpolarization occurring there as well. Okay? All right. So remember, these are voltage gated channels. So if that's happening at one point in the membrane, then right next door to the membrane, when we see these different changes, when we see depolarization occurring, that's going to affect the area next to it. And once that one starts responding, that's going to affect the area next to it, just like the repeating wave like we see in the stadiums. Okay? All right, so let's see how that comes together in an animation. Because it's one thing to see these step by step, like the sliding filament mechanism. It's another thing to actually be able to see it moving.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only, and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
So you should be able to draw that out, be able to label the axes, be able to, say where's resting potential, where's the graded potential, where's the action potential, what's depolarization, repolarization, hyperpolarization, and what's causing that, which ions are moving which ways, voltage-gated channels, things like that. That's big. So make sure that you could do, much like you see in the graph in that last little segment there. Okay. So, like we said at the very end as well, when you get back to resting potential, that's due to those leak channels and the sodium-potassium pump going again. So let me show you, in terms of resting membrane potential and this transition to action potentials, show you another short video that comes from somebody you may recognize. Let's see.
The resting membrane potential is a voltage that exists across the plasma membrane of all cells, including neurons. The membrane potential can be measured with a voltmeter that measures the electrical potential inside the cell relative to the outside, as seen here in a section of the neuron's axon. In a resting neuron, the membrane potential is about negative 70 millivolts. This negative value indicates an unequal distribution of charge across the membrane, with relatively more negative charge on the inner side of the membrane. The resting membrane potential is primarily due to two factors. First, there are different concentrations of sodium and potassium ions both inside and outside the cell. Second, the membrane is more permeable to potassium ions than to sodium ions. We'll examine each of these factors separately and then show how they work together to generate the resting membrane potential. Let's begin with the unequal distribution of sodium and potassium ions across the plasma membrane. Outside the cell, in the extracellular fluid, we see a high concentration of sodium relative to the sodium concentration inside the cell. Recall that this difference in concentrations represents a concentration gradient. Because of this concentration gradient, sodium tends to diffuse into the cell through any available pathway. The opposite is true for potassium ions. The potassium concentration is high inside the cell relative to its concentration in the extracellular fluid, so potassium ions tend to diffuse out of the cell. Both the sodium and potassium concentration gradients are created and maintained by active transport. The sodium-potassium pump uses ATP to constantly transport sodium and potassium ions up or against their respective gradients. The second factor leading to the resting membrane potential is the membrane's selective permeability. Selective permeability is a property of the membrane that allows some solutes to diffuse through or permeate the membrane more easily than others. Because ions cannot cross the lipid bilayer directly, most of the membrane's ion permeability is determined by ion channels. In a resting neuron, leakage channels are the only channels that are open, allowing ions to permeate the membrane. Although leakage channels are always open, these channels are selective. Potassium leakage channels only allow potassium ions to pass, while sodium leakage channels only allow sodium ions to pass. The plasma membranes of most cells contain many more potassium leakage channels than sodium leakage channels. This means the membrane is much more permeable to potassium than it is to sodium. Having explained concentration gradients and selective permeability, let's talk about how they work together to generate the resting membrane potential. To do this, imagine that the membrane contains only potassium leakage channels, and that, initially, no voltage exists across the membrane. From this initial condition, potassium ions would diffuse out of the cell down their concentration gradient. Because these ions are positively charged, as this process occurs, a negative membrane potential develops, where the inside of the membrane is more negative relative to the outside. This process does not continue indefinitely. Because opposite charges attract, positively charged potassium ions are attracted to the negative interior of the cell. This so-called electrical gradient affects potassium diffusion. So while potassium's concentration gradient favors outward diffusion, the electrical gradient favors inward diffusion. The sum of these two gradients is known as the electrochemical gradient. At a membrane potential of negative 90 millivolts, potassium's electrical and concentration gradients are equal in magnitude, but opposite in direction.
Isn't that exciting? So yeah, I used to do some work for Pearson, creating some of these instructional videos. So that one was basically on resting membrane potential. If you want to see more of that, if you felt like it was helpful and you wanted more of that in terms of resting membrane potential, you can let me know. I can try to post it on our course webpage. But remember, we need to know how resting membrane is established, what's involved, etc. And then what happens when we change that with a graded or an action potential. Okay, let's move on. So a key thing here is that, like when we saw with that graph, there's that time when it's starting to rise, and it rises a little bit slower, which is where we're getting up to threshold. So, you know, when we're depolarizing the cell, there's going to be a time where it's stepwise, it's kind of going up and it's becoming a little bit more depolarized, etc. But it's not until it reaches that threshold do we see the massive explosion of these opening of the voltage gated channels. And so much like the trigger on a gun, right, you can put a little bit of pressure on it and nothing's going to happen. A little bit more, nothing's going to happen. When you get to threshold, then you move that trigger all the way and the whole barrel moves, etc. So in that case, you get the massive explosion, the big change, right? So it's an all or none principle. If you get at the threshold, you get the same response every time. If you don't get at the threshold, nothing happens. And that's a key aspect as well. The all or none means that these channels are going to open up and we're going to see an action potential be generated. And the action potential, that big peak or mountain that we saw, is going to be the same height every time. It doesn't change. The response is the same every time. Like the firing of a gun, the bullet doesn't go faster if you squeeze the gun harder, right? That trigger doesn't produce any difference in terms of how the gun responds once you get to that pressure and you get to threshold, right? So it responds the same way every time, as long as it's loaded. So with this propagation of the action potential, like we said, once we create this change and we start moving particles around, then it's self-propagating, right? What we find is what happens at one area affects the next area, and they do the wave, and they do the wave, and it goes, and it doesn't diminish, right? We saw in graded potential, it diminishes. Action potential, you get the same peak occurring again and again and again and again all the way down the axon. Now in an unmyelinated cell associated with the gray matter, right, then we have to reproduce that every step along the way. But in a myelinated one, we only have to do that at the axon hillock to begin it, and then at each of those axonal nodes, at the nodes of Ranvier. So it's kind of like, you know, one of those merry-go-rounds they used to have at different playgrounds. You know, you'd have to sign a waiver to go on one now. But, you know, when some kid's on it, you want them to go around, every once in a while you'll grab it and you'll push it, and you'll push it, and you'll push it, and that will keep it going. That's what's happening at those axonal nodes, okay? So there's a lot of these voltage-gated sodium channels there, the axonal nodes, and so we regenerate that wave, that action potential, and now it's moving down, down, down the axon. And that's another thing. It can't move the opposite way, because what happens is those channels that were just active, they go into what's called a refractory period. And a refractory period means that they cannot respond for a period of time. All right, so let's take a look at that. For now, I guess because, you know, these are pretty complicated steps, instead, let's pause here and we'll start with this video next time so that you can have a chance to go over it yourself and then come back and we'll hit this topic one more time.